right, I think we'll go ahead and get started on this next Ivan NMR research workshop, Tracking Real-Time Proline Hydroxylation in IDPs. This time I'll turn it over to David Rice to get us started.
So why don't we get started? And our speaker is Geneviève Seabrook, who will be talking about hydroxyproline.
Well, thank you very much. I would like to thank the event team for allowing me to present some of the work that we are doing here at Princess Margaret. And I think this platform is definitely appropriate for the work that I want to show. It'll be a little bit casual, not super formal, but you'll understand why. So the title of the talk is Tracking Real-Time Proline Hydroxylation in IDPs. And but the title of the talk could have been really selecting the most appropriate NMR tool. So whether you are in a chemistry lab or a bio-NMR lab, you know, we all have multiple frequency magnet. You know, we also have a multitude, or well, maybe not a multitude, but we also have several probes, whether they are proton optimized, carbon optimized, nuclei optimized, whether they are cold probe or room temperature probe. We also have the receivers. You could have one, you could have four. Speaking about tools again, also you could do solution NMR, or you could do solid state NMR. You could also do in-cell NMR. So, I mean, for all this, we have lots of tools, whether it's the toolbox to fix your instrument, or all these tools that I'm mentioning right now. You also have on top of it, you also have the pulse sequence. So if you look at your libraries, you have tons of pulse sequences. But when you work on a project, you really need to use the proper appropriate tool. And this is what we're going to be doing for the project I want to mention. So in this case, I had to use the 800 because we're going to be working on, we're going to be talking about IDPs. I had to use carbon detection because the amino acid of interest is actually proline. I had to use multiple receivers because I'm a busy person and I don't have time to set up multiple experiments. So I want to optimize my time, and dual receiver was the best thing to do. And then, of course, I selected the pulse sequences that I needed for to answer the question of the project. But just a little bit of history, and of course, it's not the whole story, but in 1956, Paul Loutherberg introduced some direct detection of C13 NMR. And so he published the data. This was done on some small molecules. And maybe within a month later, Holm, Lundy, and Smith also observed C13 chemical shift, but also spin coupling in chemical compounds. So these were published in early 1957. Then in the 90s, direct observation of C13 resonance was introduced for organic molecules at natural abundance, obviously. However, the low sensitivity of the carbon, the low natural abundance, and the low gamma led to very long acquisition time. So it's clearly not ideal. But anyway, they were able to observe directly carbon C13. Then the biomolecular NMR world started really in the 80s and 90s, where they started developing a multitude of pulse sequences, relying exclusively on proton detect experiments. So I didn't put the names because there are so many, but still, at that time, C13 direct observation was definitely not considered practical. However, the improvement of FT, Fourier transform NMR, isotopic enrichment, cold probes made heteronuclear direct detection possible. It opened basically a new door. But that's not it. Early 2000, the first dedicated C13 direct detection for protein, mainly IDPs, IDRs, made its way. The pulse sequences for protonless carbon-detected biomolecular NMR made its way in all the libraries of the publication. But Dodge in 2000 and all demonstrated protonless high-resolution NMR for biomolecule using C13. Wolfgang Bermol from Brucker with the group of Bertini and the others introduced advanced C13 direct detection experiment for backbone assignment of prolines. And again, this group was mainly working, and the target were really the IDPs, intrinsically disordered protein and regions. And then in 2006, 2008, during that time, Bermol, Piratelli, and Isabella Fellini, they developed more pulse sequences where they could that they could use to assign to do the backbone assignment, but also the sidechain assignment, again for the IDPs and all that. So this method explored the advantage of C13 detection in case where the proton exchange or relaxation are not ideal. But in the 90s, 2000, at the same time, the use of multiple receiver was starting and making its way. And I completely remember going to the ENC a couple of decades ago and seeing Eric Kupse at his poster and talking about the use of multi-receivers. And I remember at that time, a couple of decades ago, I was really wondering how on earth I could use that for my own experiment. And here we are today, and I will talk about that. So then in 2016, Edscon Group developed the concept of utopia, which is unified time-optimized interleaved acquisition NMR for unified acquisition of standard high gamma, such as proton, fluorine, but also low gammas for C13 and 15 and phosphorus detected within the same experiment. And in his paper, he's only using one receiver. But when you put all those tools, those ideas together, you can actually accomplish something that is called, that combine the utopia idea and the use of multiple receiver. And it led to the CONHSQC. So there's a group at the University of Toronto. They contacted me a few years ago, and they're working on Hypoxia-Inducible Factor HIF1 alpha, and they were interested in looking at the hydroxylation of two prolines. And they came to me and said, oh, you know, could we use could we run an HSQC and see if we can follow the hydroxylation of those proline? And I said, yes, sure, we can do that. However, I'm not sure I don't think it's the best option. And I will explain why shortly. So just to introduce HIF1 alpha, HIF1 alpha is a protein that gets hydroxylated in presence of prolyl hydroxylase. This enzyme hydroxylate HIF1 alpha at two proline sites. When it's hydroxylated, E3 ligase VHL can form a complex, interact with HIF1 alpha, and then mark it to ubiquitinate it and mark it for degradation. At the same time, factor inhibiting HIF1 alpha can also hydroxylate HIF1 alpha at an asparagine position. In this case, HIF1 alpha is unable to interact with the co-activator P300 and form the complex. Now, in order for those two enzymes to work, they need some cofactors. So iron, the 2-oxoglutarate, and they of course need oxygen also. However, their enzymatic activity can be repressed by some oncogenic metabolic intermediate, such as the 2-hydroxyglutarate. So on the other hand, in a hypoxic environment, those two enzymes are inhibited. They cannot function. Therefore, HIF1 alpha is able to translate to the nucleus and then interact with HIF beta and form a complex with the coactivator P300. When those three interact, they are able to activate transcription of the HIF target genes. So that's for the story of HIF1 alpha. So we're going to concentrate. What I'm going to show is more the the kinetic of this reaction of PHD2 with HIF1 alpha. So very, very quickly, HIF1 alpha domains, there are three main domains: the dimerization domain, the DNA binding domain. So that's this domain over here. I don't know if you can see my arrow. Can you see the arrow? Or no?
Yes, we can.
You can. Okay. All right. So, so we've got the dimerization domain, DNA binding domain. Then we have a proteasomal degradation domain, which has the disordered region, and then it has a transcription domain. So if you use AlphaFold, it predicts that you've got some secondary structure element, but mainly huge, large loops. So the segment we're going to look, the region of HIF1 alpha we're going to be looking or talking about is the one that is this colored in red. And you can see it's a very huge, long, disordered region. So, and for the people interested also in how the hydroxylation is happening, you've got your prolines, and then with the 2-oxoglutarate and in presence of oxygen, this carbon, the carbon gamma of that proline gets hydroxylated. So you can imagine that it can really change the environment of your protein if it's folded, but in this case, it's not. So for the people that are not familiar with protein, I just showed the HSQC of a GTPase, a very nicely well-folded protein. So you can see row A is the proton HSQC has a spectrum with a very nice dispersion. But now, if you look at HIF1 alpha, which is disordered, a disordered region, you can see that the amide dispersion is very poor, and you've got quite a large overlap of the amides. So can you follow the hydroxylation of this disordered region using the proton and 15-HSQC? The answer is yes. Of course you can. It's not an issue. It's not a problem. And actually I show right here that in black is the spectrum before hydroxylation, and in red is the spectrum after hydroxylation. So in this case, you can actually follow the amides peak shift. But in this case, of course, you're not going to follow your prolines because there's no NH group. So you're going to rely on reporters. So for example, this alanine 403 is right after the proline 402 that gets hydroxylated. So yes, you can follow those reporters. You also have the tyrosine 565, which is right somewhere in the middle there and gets shifted also quite far away, but it gets shifted in a very crowded region. So can we do it? Yes. Is it simple? Not so much. Therefore, if you have an 800 megahertz, for example, you can get a beautiful dispersion of your carbons, and you can also use a carbon direct optimized probe. Therefore, you can run the CON experiment. And here on the right is the spectrum of the CON in this dimension. You've got the carbonyl, and in the other dimension, you can observe the nitrogen. So you can see the black spectrum is before hydroxylation, and in red is after hydroxylation. And again, you can see the peak shifting for the reporters that we could see already in the HSQC. But much, much, much better than that. You can observe the prolines. Now you are observing directly the amino acid that's getting modified. In this case, it's the prolines. So now we can see the prolines, which we could not see in the HSQC. But the other advantage is that the chemical shift for those proline is extremely well placed in the sense that it's quite far away from the rest of the other amino acids. So it's very well separated. So we've got multiple advantage by using the carbon detect CON. The first one is you have an excellent chemical shift dispersion. The second one is that you can observe the proline. And the third one is the fact that this region for the proline is well separated from the rest of the amino acid. So in 2 minutes, well, once your experiment is done, in less than half hour, you know immediately which prolines are getting hydroxylated, which is really, really neat. So I'm going to show the kinetics, and basically I will show the zoom of this region here. So we will follow the position of those two proline when it's not hydroxylated and when it's getting hydroxylated. And that's what I'm going to show. So our student, Wen, was able to assign all those residue in the IDP. He also plotted the chemical shift difference between before hydroxylation and after hydroxylation. And it's, for the people who work on protein, it's super interesting because we're so used, to see some amide being affected and others not. Here, it's very interesting because pretty much nothing is getting affected except the two sites where you have the two proline, and that's for the carbon beta, carbon alpha, the carbonyl, and the NH groups. So, but pretty much everything else does not really get affected. So it's quite a huge difference. Of course, we do expect that, but you could see some differences also. So the one question was, what is the affinity of the enzyme for those prolines? So the student was able to use BLI, and using BLI, he did show that those two proline have the same affinity for the enzyme, or the enzyme has a similar affinity for both prolines, since the case is very similar. So here is the kinetic. You can see here time 0. You've got your two proline, the proline 402 and proline 464 at time 0. Less than an hour later, you can see the first proline is already shifted. And then it takes another 7 hours to start seeing the second proline getting hydroxylated. And it takes about 19 hours to get both proline fully hydroxylated. So there's clearly a difference between the two sites, basically. So on the right, you can see that proline 564 gets hydroxylated extremely quickly, extremely fast. Then proline 402 in red, the red circle, you can see the hydroxylation is taking place, but it's just super slow. And both reaction goes to completion. Now, the idea was to compare, well, what will happen if we mutate one of the prolines? So in the open circle, we mutated the proline 402, and you can see that the rate of hydroxylation for 564 decreased a tiny bit, not so much. And now, if you mutate the second proline, 564, to an alanine, it definitely does affect the hydroxylation rate of the first proline 402. So with this data in hand, we could tell that the hydroxylation efficiency of P564 definitely predominates the one over 402. So the question was, okay, well, why is this happening? So I guess it was known that this enzyme, PHD2, had a recognition motif like the leucine, XX, leucine, alanine, proline. If you look at the two sites, you can see it's pretty much conserved among species. So you do have the recognition motif on both sides. So the question is, okay, why 564 is getting hydroxylated a lot faster? So we decided, okay, so let's look at the difference. The difference is that the amino acid that is following proline 564 is a tyrosine. Compared to P402, you've got an alanine. So we decided, okay, let's mutate this tyrosine to an alanine and see how it affects the hydroxylation of P564. And then let's mutate this alanine, the alanine that is following P402, to a tyrosine and see if we see any changes here. So if you look at the rate here, you can see that when the tyrosine, this tyrosine that is conserved pretty much everywhere, if you mutate the tyrosine to an alanine, the rate doesn't change much. But now let's mutate the other alanine to a tyrosine for this proline over here. Well, you can see that the rate is now increased somewhat. So it is clear that that tyrosine contributes to some extent to the efficiency of the prolyl hydroxylation. So we went on and we did a domain swap where we actually completely swapped the whole domain, these two domains. And again, so you can see on the right the overlay of the HSQC before hydroxylation and after hydroxylation. And you can see that you get pretty much similar data, similar spectrum, sorry. And then in here, what happens if we compare the rate? So the black circle corresponds to the kinetic to the wild type for proline 564. Now with the domain swap, again, the rate of hydroxylation for that proline did decrease a little bit, somewhat. And that's a reason, a consequence of that domain swap. However, for the 402, the proline 402, when you do the domain swap, you actually you lose a little bit more of the rate decreases in that case. So the amino acid composition of the second domain contributes to the prolyl hydroxylation efficiency. So we're definitely not done with the study of trying to understand what's going on really with those two prolines. But anyway, the way I set up those experiments at first was that I would collect an HSQC and then a CON, and then an HSQC, a CON. So I intercalated the CON within those HSQCs. Well, it is actually time consuming. And I thought really pretty quickly that I did not believe it was very efficient. And then I thought we should combine those two experiments. And sure enough, I was at some meeting at the ENC and a few years back. And then sure enough, Isabella Feli showed a spectrum where they had combined the CON and the HSQC. So we had the same idea. And I remember telling Bob Bernot that that's what I wanted to do. And then, bong, she just presented a few minutes after. So how does it work? It's very simple, actually. The idea is very, very simple. So you've got first your HSQC, and then right behind, you've got your CON, the scheme for your CON. But the way it's going to work is that you're going to use a first receiver to collect the proton magnetization, the proton signal, sorry. And then you're going to use a second receiver to collect the carbon signal. But you do have to be careful. You have to make sure that your D1, your relaxation delay, is long enough so that all your carbon magnetization can come back to equilibrium before you start your second HSQC. And on our instrument, the way it's set up, we use the terminology of parent and child, like described in that Utopia paper, where, so on channel 1, you've got the proton, and then you use the receiver 1 and you collect the proton signal, and then you set up the child experiment. And so channel 1 is your C13, and then you're going to use another receiver to collect your C13 signal, and then that's what you collect here. So in this experiment, you collect these NHs that you get when you run a simple HSQC, and you also collect the correlation between the carbon and nitrogen in same as you would get in your CON experiment. So why to do that? Well, the advantages are that you take advantage of the relaxation delay. So that's the one advantage, and also multiple receiver. You might as well if you've got them in your console. The way you set it up is that you set up your HSQC, like exactly a normal HSQC, like you would do all the time. And then there's that icon here, the M. You click on it, it pops up another window. And then in this window, you set up your CON experiment. You do have to be careful with the delays and all that, so you need to adjust those delays. But the one delay that you need to really, really pay attention is the delay that's going to set up the SW dimension for the nitrogen. At the beginning, I had some issues with that because I did not realize that that delay had to be adjusted. But anyway, once I found out, then it was all very straightforward. It is actually pretty straightforward to set up this experiment. So here you can see one receiver versus two receivers. On the top left, you've got the normal HSQC, 10-minute HSQC. With this concentration, I think we could have done even a 5-minute HSQC, would have been still very good. The CON at the beginning, especially when I was starting dealing with carbon detection, and also the dual receiver. In this case, the acquisition was 45 minutes. I used 8 scans, and the relaxation delay was 1 second. And I left it there because I was able to see all my carbon. So, but of course, if you're missing carbons, you better make sure that you have a long enough relaxation delay. And then if you overlay the CON that is acquired by itself versus acquired in that CON HSQC type experiment, you can see that the overlay is pretty similar. The advantage was that I was able to get an acquisition time of 25 minutes and run only 4 scans. This is super critical when you're doing a kinetic. You want to get most of your point at the beginning of the kinetic. So even 25 minutes is kind of too long. I didn't get a chance to try with 2 scans, but maybe it's something I can try next time. So, like I said, it's definitely a benefit for the kinetics. And also you set up 1 experiment instead of 2 experiments. And here is the result for using that CON HSQC experiment. You can see the overlay again of those 2 spectrum before hydroxylation, and then at the end of the hydroxylation, you can see the 2 proline clearly right away. Immediately, you can see those 2 proline shifting a field. And in here, on the right, you have time 0. You have your 2 proline. 30 minutes later, P564 shifts already, start shifting. After 1 hour, P564 is completely hydroxylated. It's gone. And then it takes another 6 hours later to start seeing the proline 402 to get hydroxylated. And it takes a lot longer for that proline to get fully hydroxylated. So the advantage of this experiment is that you get complementary information. So would I go and just run one of the 2 experiments? No, because I am always thinking, oh, I may miss some critical information if I don't see my NH groups. And sure enough, actually it is true. I also needed to run that experiment. But the goal here is that you are interested in prolines. The prolines are the ones that are getting hydroxylated. Therefore, you should pick an experiment where you can directly observe the amino acid that is getting modified. In our case, the CON was the perfect choice, was the excellent choice, actually, again, because of that nice dispersion, and also it's separated from the rest. And immediately, in a very short amount of time, you know immediately which proline are getting hydroxylated. Of course, this methodology can be applied to the world of IDPs and IDRs, obviously. It can be also useful if you have a sample that have a very short lifetime. You can apply this methodology on cell lysate and whole cell. And of course, you can apply this experiment to other post-translational modifications. So just to go back to the beginning, this was really about, how NMR managers, NMR, whatever, we're super busy. We have lots of tools in our core facility or NMR center. We have lots of projects, and we don't have enough time to explore everything. But this one was a beautiful demonstration as to which instrument you should be using, an 800 because you need dispersion. Which probe should you use? Of course, the one that is optimized for cystatin. And also, why would you need two receivers? Because, well, that's going to save me time. And believe me, we don't have much time. We're always super busy, and so on. So it's all about the fact that we all have NMR tools in our NMR lab, not just to fix the instrumentation, but also to apply the proper tool to the proper project. So in here, why doing cystatin direct detection? It's mainly because it's a complementary tool to proton detect. I'm not saying we should stop doing proton detect. Obviously, we need it. It's critical. This cystatin direct detection is important for disordered protein because you've got a much better dispersion. It's, of course, useful for the region that, for whatever lacks proton, well, in this case, the prolines, but also the carboxyl groups. It's also useful for high salt and also exchange product environment, and also for paramagnetic system with low relaxation loss. And what's the advantage for the multi-receiver is the fact that it's pretty neat because simultaneously you do the acquisition of signal from two nuclei, two of them. So it's pretty cool. I really like that aspect. You do a parallel acquisition, so that definitely reduces your total experiment time. You also improve the sensitivity and efficiency because you are using all the available magnetization. So if you run two experiments separately, you queue them while you're going to lose some magnetization during all that time. Also, you can simultaneously observe heteronuclear without switching the hardware. Well, so this is true for the type of console that I have in my lab. But one time I was presenting a similar work and someone asked, oh, do you have to have a special console for that? You probably could do it, but you would need to switch the hardware in that case, if it's an old console. It's also cost effective because if your experiment is long, you need to charge more for the core facilities. But, often we've got instrument and we need to get going with those experiments. So you want to reduce the instrument time. So these are the advantages of the cystatin direct detection and the use of those dual receivers. Honestly, I was a bit worried, a bit scared about using multiple receiver, but when I started, it was like, oh, this is so cool. We need to continue. So just to finish up here, I'd like to acknowledge the PhD student from my co-host lab, Wenhee. So that was his project, and I helped him for the NMR part. So I'm going to end right here, and if you have any question, I'll try to answer. And thank you for your attention.
Thanks very much, Geneviève. So I'll ask everyone, are there any questions?
Yeah, good evening. My name is Siddhartha. I'm from Bangalore, and I'd like to ask a question.
Would you like to display your
Yeah, just one second.
Yeah, okay.
I'm having trouble. There we go.
Okay.
Okay. I don't know if you all can see me now.
Yes.
Yeah. So, Dr. Geneviève, that was a very interesting talk. I would like to know whether did you observe any prolyl cis-trans isomerization when you did this? Were you able to track that down?
It was a bit muffled. So did you ask about the prolyl isomerization?
Yes.
Yes, okay.
Yeah.
So yes, we actually did look into it. Let's see if I can show. So I can show it in here. So we did look into it and we really follow. So if you look at, so I didn't talk about it. I didn't put it in here because it was it was not conclusive. So that's why I did not put it in. But if you look at the HSQC, we have some very weak peak with weak intensity. And this corresponds to the cis conformer, isomer. So, and also we see some very weak peaks with the CON that we didn't get a chance yet to look into further. But it was clear that if you only had the trans, the reaction was not changing. The rates were not modified.
I see.
Yeah,
Because
we did look into it, yes.
The cis-trans isomerism is actually better monitored by the difference in the C-beta, C-gamma chemical shifts of proline. So if you take the delta-delta, C-gamma minus C-beta, if it is 4 to 5 parts per million, then it is trans, but if it is more than 8 parts per million, it's actually cis conformer. So it's very easy to track from the carbon spectra, but you'll need like a proton-carbon HSQC or a HSQC toxi-like experiment for that.
So I have done it. I have actually done that. I've done even better. I have run the sidechain experiment with the carbon detect, and you clearly see what you were describing. Exactly.
Okay.
Yeah. Yeah.
And may I ask another question? The PHD2 enzyme, did you clone and express that enzyme?
Did I what? Sorry? Did I clone?
Do you have a clone for that to express the enzyme, the prolyl hydroxylase?
I would have to double check with the group whose project it is. I'm trying to remember whether the student also made the enzyme or not. I would have to double check with the student, but I don't know. I would have to double check with him, but I know he's been purifying enzymes. So PhD2 may be one of them.
So, so would it be okay if I wrote to you and followed up on this question?
Oh yeah, absolutely. Yes.
Okay. Thank you very much.
Actually, I'm having a meeting tomorrow morning with the PI, so I can ask that question then.
So my interest in this is because I work on animal toxins, many of which have this proline hydroxylation.
Oh, okay.
And yes, and I've actually cloned and expressed many of the peptides, disulfide-rich peptides, and I'm trying to do the proline hydroxylation, but I'll have to purify the enzyme from natural sources like chick embryos, which is a little tedious actually. So that's why. Anyway, I'll write to you. I won't hold up the meeting. My selfish interest. Thank you very much. Yeah.
Okay, you're welcome.
I see that Alexander has his hand up.
Oh yes.
Could you show yourself?
I think I turned on my video. Maybe this. Oh,
okay, that's all right.
Oh, sorry, it was the
camera.
There we go. Oh yeah, you mentioned the side chain experiment. So is there actually a better way of correlating them? Maybe C delta or C gamma with nitrogen? Because I think the hydroxylation is at the C gamma position, right? Or the carbonyl shift is sufficient.
Yeah. What is the question? Sorry.
Oh, you're saying, I think the hydroxylation happens at the C gamma position, right?
The proline, yeah.
you did the side chain experiment, side chain detection. Do you have like better sensitivity to this hydroxylation step when you look at the C gamma or maybe C delta rather than carbonyl?
No, the carbonyl were much better.
Works much better. Okay. And another question, kind of somewhat plugging our own equipment, because down here at the University of Georgia, we have a 900 and a 1.1 gigahertz with the TXO cryoprobes. Would that be of benefit going above the 800?
Well, for the dispersion, yes. So what I used is very similar. So it was 800, not 900 or not 1 gig. But I did use my TXO cryoprobe. And that's how I was able to run those experiments. So I do believe that, yes, indeed, it'll be even better. I mean, if you go to higher field, you'll have better dispersion of your carbon-nitrogen correlations. And the TXO, yes, of course. That's, well, I mean, it depends. If it's carbon optimized, yes. If it's nitrogen optimized, then no. So it depends which one you got.
Yeah, okay, I need to double check. Although nitrogen detect experiments might be useful too under certain circumstances, I think, with IDP.
Yes, absolutely. Yeah.
But the ones you had was just dual channel, right? Is it, or it's also a TXO on the 800?
It's a TXO.
A TXO, okay.
Yeah. One of the 800 has a TXO cryoprobe and the other one has a TCI cryoprobe. So one is optimized for carbon, the other one is optimized for proton.
Oh, okay. Yeah, obviously, because you did simultaneous with all three channels, right?
Yeah.
Okay, thank you.
You're welcome.
Okay. More questions? May I ask a general question?
Yes.
So in talking about, we have a new, actually only a 600 Bruker with the optimized carbon, but we haven't figured out how to use it yet. So how much larger in general can you go in proteins, maybe even folded proteins, when you go to carbon detection?
Oh. That's
maybe that's not an obvious question to ask.
No, I think you can go pretty high because most of the time your carbonyl, you can observe your carbonyls a lot better than anything else.
Okay.
So, but how high in molecular weight?
Mm-hmm.
So for
Well, you don't have to be specific, but
right. No, but I don't know. I haven't done the huge complexes yet. So this one was no, but it's a very interesting question. The question is, it's the higher molecular weight. I mean, it's still pretty good because if you run the CON on a high molecular weight of a disordered protein, you still will have, of course, that's assuming you go to a high field, you will still have a beautiful dispersion. So I don't see a problem. Now, okay. So I played around with carbon detection when I had a TCI probe. That was years ago, and I didn't like it. So I decided, okay, next upgrade, I'm getting a TXO probe. But I did play around with when I had a TCI probe, and I practiced in quote, on a folded protein. And the result were a lot better on disordered protein than folded protein.
Right. Yeah.
And of course, it's the correlation rate that is definitely a factor, how tumbled and all that. So, but I think, well, I mean, when I have another project, so all the IDPs that I worked with, they were less than 30 kilodalton. So, and it worked for all of those. So 30 kilodalton is definitely feasible. So, but I don't see why you couldn't do it with a higher molecular weight for IDP or IDRs.
Okay. Okay.
Yeah.
Well.
That would be interesting to try.
Mhm. So are there more questions? So if there are no more questions, John, are you there? Would you like to say anything? Okay, so with that, I want to thank Geneviève for a great talk, and with this, I'll close our meeting. I don't have the gavel handy, so.
Thank you very much, Dave.
Okay,
Thank you,
thank you.
everyone. Thanks.
Okay,
Thank you.
Bye-bye.
bye.